Live from the North Carolina Museum of Natural Sciences in beautiful downtown Raleigh. It's the Science Cafe.
Good evening, everyone. Welcome to the North Carolina Museum of Natural Sciences. My name is Chris, and I will be your host for the evening. So welcome to the Science Cafe. We are live inside the Daily Planet Cafe, inside the Nature Research Center at the North Carolina Museum of Natural Sciences, which means there's a lot of awesome inside a lot more awesome, inside even more awesome, and it's right here in the middle of downtown Raleigh, North Carolina. I'm so glad we have a huge crowd, so I am thrilled that so many people could make it out to the cafe tonight to meet and hear from what I think is going to be a pretty exciting presentation. We have here with us tonight Dr. Gavin Williams from the Department of Chemistry at North Carolina State University. He'll be talking to us about lots of really interesting things. In getting ready for tonight's program, I was looking over some of the materials. I had his slides beforehand, so I cheated just a little bit. And I was seeing some things that were really catching my attention and piquing my interest. Terms like synthetic biology and Legos, which made me very excited. Both of those, not just the Lego part. So with that, I will go ahead and hand it over to a gentleman whose accent I think you will love because he's not from North Carolina originally from the UK, and I will hand it off to Dr. Gavin Williams.
Thank you very much for that excellent introduction, and thank you everybody for coming out here tonight to listen to me talk. I'm going to spend the next 20 minutes or so describing to you my research group's efforts towards engineering microbes for the synthesis of new drugs and other small molecules. And we do this by using a philosophy, a way of doing science referred to as synthetic biology. And one of the things that I'm going to do this evening is describe to you what synthetic biology is and how we use it in the lab, and how ultimately, in probably just a few years' time, synthetic biology will essentially permeate almost every realm of your lives in society. What I also might do is explain why there's a picture of me as a 5-year-old playing with Lego bricks on the floor of what I presume was my house somewhere in England about 30 years ago. And so what I'm going to argue during the next 20 minutes is that I was actually much more intelligent than that picture implies, and that I was already at 5 years old preparing for my life and career as a synthetic biologist 30 years later. So my group and others, our goal is to engineer microbes. And usually microbes get a pretty bad rap with several good reasons. So this slide shows a few agar plates with some interesting microbes growing on the surface of these plates. The first one on the left is called Staphylococcus aureus. This is the parent strain responsible for MRSA, MRSA, methicillin-resistant Staphylococcus aureus. This is a strain of bacteria that might be very familiar to you. It causes boils. I didn't want to show any pictures of these because I know some of you might be eating this evening. But this is a really, really serious bacterial infection. This is not a very nice microbe. The next one is called Bacillus anthracis. As the name suggests, this is a bacterium that produces anthrax, which is actually the spore form of this microbe. And so you might be familiar with anthrax. Many governments across the world are intrigued about using anthrax as a biological warfare agent. But the two microbes on the right, I posit, are microbes that you may have not heard of before. The first is called Streptomyces griseus, and the one on the right is called Streptomyces coelocola. These two microbes are actually enormously useful to humanity. and maybe you haven't heard of them before. So the two microbes on the left are definitely our foe, but the two microbes on the right are definitely our friends. And what makes them our friend or foe are the variety of molecules that these microbes produce. So microbes produce a variety of very large molecules like proteins and peptides, and a variety of much smaller molecules which we refer to as natural products or secondary metabolites. And it's this profile of small molecules and proteins that microbes make that ultimately determine whether they're good for us or bad for us. So this is an example of a few small molecules that are made by the microbes which are our friends, Streptomyces, as they were described in the previous slide. So these are complex small molecules that are biosynthesized using the metabolic pathways inside these microorganisms. You might not recognize the structures of these molecules, but unfortunately, some of you might be familiar with these products. So the first one is penicillin. I think we've all heard of penicillin. This is a game-changing antibiotic that was first used in World War I, and it really changed the way that we were able to treat patients in hospitals. The next example is called Mevacor, otherwise known as lovastatin. This is another example of a molecule that's produced in nature inside microbes. And some of you might take lovastatin to lower your blood pressure through clearing cholesterol in your bloodstream. The next example is called Rapamune. That's the trademark from this Pfizer compound. This is another example of a natural molecule that's produced inside microbes. In this case, Rapamune, also known as rapamycin, is used in donor recipients during transplants to reduce the immune response to the transplanted organ. Absolutely critical small molecule that's used to revolutionize organ transplants. The next one is called ivermectin. This is a really beautiful structure. Again, another small molecule produced by microbes in nature. Some of you might be familiar with ivermectin. It's the active ingredient of HeartGuard, which we give to our dogs and cats to prevent worms and other infections inside their bodies. So I give this to my dog every month without fail. And then finally, the next structure is another example of a complex small molecule produced in microbes. In this case, this particular molecule displays very potent activity. It kills insects, and many of you might happen to spray this in your backyards. And so really what this slide illustrates is the really powerful activities of these small molecules which nature has already produced for us. And these microbes are a really tremendously powerful source of new molecules that we might be able to develop new drugs from. These particular examples are all molecules that nature has already produced. And these are the active ingredients of these drugs, and these molecules have not been modified at all from their natural forms produced from the microbe. These are rare examples. Normally, chemists and medicinal chemists have to alter the structures of molecules like these in order to optimize the biological activity or to fine-tune the pharmacological properties of these molecules before we can use them as drugs. And so very often we need to come up with ways to alter and diversify these structures. There's another reason why we have to figure out how to alter and diversify the structures of these molecules that nature already provides. And one of the most compelling arguments is the urgent need to overcome antimicrobial resistance. So we are in a real dire, urgent need to create new antibiotics to replace the ones which microbes that infect us have already evolved resistance towards. So right now, across the world, 700,000 people die each year from various drug-resistant infections. And it's projected that by the year 2050, if we don't do anything about this situation, 10 million people will die every year due to drug-resistant infections. That's the staggering number of deaths due to infections. And along the way, up until 2050, it's estimated that the cost to society is going to be in excess of $100 trillion in order to cope with this massive amount of resistance and deaths and illnesses. This might affect everybody across the whole world. And in one example of a drug-resistant microbe, Klebsiella pneumoniae, this is something that was actually first detected right here in North Carolina, possibly in a hospital in Raleigh. And in under just a few years of this antibiotic-resistant strain being identified in North Carolina, it was found in multiple locations across the world. And that's just one example of one drug-resistant strain that was found in our state. So this is a really, really important area. We really need to figure out how to produce new antibiotics, and we need to figure out how to diversify molecules which are already produced by microbes in nature. So where could new antibiotics come from? Well, I've already described that one way that we can afford new antibiotics is to take the structures that nature has already made and figure out how to diversify them. We can learn how to modify the chemical structures of antibiotics that are already found in nature, biosynthesized in microbes. And this is one example of an antibiotic that we like to study in my research group called erythromycin. And it's spinning around maybe a little bit too fast. And you can see really that this is a very, very complex structure that we have to figure out how to diversify some way. And there's lots of other ways of finding new antibiotics. And there are other ways of altering the structure of molecules like this. But the method that my group really enjoys using is to figure out how nature made these molecules in the first place. And then if we understand how nature makes these molecules by analyzing the genomes of these microorganisms, we can then begin to figure out how to redesign the biosynthetic pathways that already made these molecules inside the microbes. And in this fashion, if we can redesign those pathways, we might be able to produce new structures that display optimized antibiotic activity. And so this is what I'm just going to elaborate on in the last of my few slides. So in some ways, the molecules that I've shown you are constructed in nature in a very similar way to how cars are assembled in modern day factories. So cars are also very complicated, and they're made up of hundreds of smaller parts. And obviously, these parts aren't just randomly assembled to afford a fully built up car. They're actually assembled in a highly logical, sequential fashion from a chassis, which is just a bare bones scaffold of a car, to the fully built car. So a car is assembled part by part. And cars are actually assembled in an assembly line fashion nowadays with incredible high throughput. And these assembly lines start at one end with the chassis, for example, and then the chassis is moved along the assembly line step by step, and at each robotic workstation, a different part of the car is selected and attached onto that chassis so that the car is built up in pieces. And the beauty of this assembly line strategy is that this assembly line is highly versatile. It's modular. So a particular car factory assembly line isn't restricted to only making one type of car. because you can program each of those robots to select different kinds of parts, and you can produce any kind of car that you want using that assembly line. So I'm explaining how cars are assembled because it's highly analogous to how many small molecules are constructed by nature. So nature's assembly lines look a little bit different from car assembly lines. I realize that there might be some symbols on this slide that you can't read from this distance, but it doesn't really matter. So each of these circles or spheres on this slide represent an individual enzyme which is catalyzing a very particular chemical reaction. And in this particular example, each of these enzymes, each of these spheres are indicated in a certain order. And so altogether, there are about 25 enzymes in this enzymatic assembly line. And altogether, those 25 enzymes are catalyzing the selection and condensation, if you will, of 7 small molecule building blocks. And so those small molecule building blocks are just shown underneath the title on this slide. And those 7 molecules are selected by this enzymatic assembly line in a very logical, ordered way to provide one single product, which is an antibiotic from this particular assembly line. I'm just going to show how this works in a little bit more detail. I just want to note that I've color-coded these individual enzymes, and you can see that they form groups of the same color, and we refer to these as modules. And you can think of each of these modules, there are 6 of them altogether, as each of the robotic workstations in the car assembly line I showed you on the previous slide. So this assembly line begins with the molecule that's highlighted in blue, which is loaded onto that blue loading module of enzymes. That's the first set of spheres in this assembly line. And then what happens is that that molecule is passed along to the next sequence of enzymes in this assembly line. And so module 1 is then responsible for grabbing a small molecule building block from the cell and catalyzing the condensation of that molecule with the one provided by the loading module. And as this growing chain of atoms is passed along from one module to the next, the chemical structure is extended by 2 carbons after each round of processing through each of those modules of enzyme activities. So this is entirely analogous to how cars are assembled. We're building up a very complex molecule sequentially by passing it through this enzymatic assembly line. And at the very end, this linear structure becomes cyclized to afford this cyclic structure. I want to point out that there are some little squiggly lines attached to these assembly lines. These are so-called prosthetic arms. So the spheres are enzymes. These are each a protein, and the squiggly arms are basically a molecule that's been attached to that protein, which acts as a robotic arm. And I'm going to show you a little bit of a video on the next slide, which highlights how one of these modules of enzyme activities actually operates during a catalytic cycle during this enzyme line assembly of an antibiotic. I'm going to highlight, I'll try to highlight where that robotic arm is on this structure. So this is a video I stole from Martin Schmeng's group at McGill University in Canada. This is the resting state of the enzyme. That protein that you just saw highlighted in blue, that's where the robotic arm is attached. So now you can start to see that blue protein start to move around. And as it does so, that robotic arm inserts itself into the orange protein. And now that rotates almost 180 degrees, you can just see that robotic arm inserting itself into the purple enzyme active site, where it catalyzes some really neat chemistry. And once it's done one cycle of that robotic arm inserting into those two active sites, it resets and starts over again. And this is the beauty of enzyme catalysis. They'll just go on forever, converting starting materials, building blocks, into more complex products and structures. So this is a really fascinating insight into how these enzymatic assembly lines actually go about their job and building up complex small molecules. And so what that video really does is highlight the complexity of how this giant enzyme assembly line takes those 7 small molecules and assembles them into a more complex structure. And bear in mind that each one of those color-coded modules is like the structure that I just showed you on that video. This is an incredibly complex molecular machine which is coordinating dozens and dozens of individual chemical reactions to make a really important antibiotic structure. And so at this point, what I want to highlight is the color coding on this assembly line. And perhaps you might be able to see that it's the identity and sequence of the enzymes in this assembly line which are a template for the synthesis of the molecule shown at the end of the scheme. We don't need to know what each of the abbreviations and each of the enzymes are doing, but if you focus, for example, on the green portion of the assembly line, that part of that enzyme assembly line is responsible for all the chemistry that's highlighted green in the final product that's made by the assembly line. So there's a beautiful relationship between the enzymes in that assembly line and the structure of the molecule it produces. What this infers is that maybe we can change the order or identity of the components in this enzyme assembly line in order to produce new types of molecules that could be powerful antibiotics. And how we do this is by going back to my very first slide where I showed you me as a 5-year-old playing with Lego bricks in my house. And so what we like to do is simply treat these enzymes, the components of this assembly lines, as if they were Lego bricks. And so if we think of them as Lego bricks, we can just take these individual bricks and start attaching them together in a different order. Maybe we can start including in this assembly line Lego bricks that aren't included in this naturally occurring enzymatic system. And by treating them as Lego bricks, maybe we can start to make really powerful new antibiotics. So I call this the Legalization of biology. That wasn't meant to be funny, but great. And what I'm going to highlight here is really the essence of synthetic biology, which is a phrase that I might have mentioned earlier on. So the essence of synthetic biology is that we can design things via DNA. We can build new things in biology by utilizing DNA, and then we can test those things by asking questions, did we make whatever we wanted to make? And so in the context of what my research group does, we might want to start off with an enzyme assembly line that already exists in a microbe that produces an antibiotic. Then we can design new versions of that assembly line by treating the individual components as Lego bricks. We can build those new assembly lines by harnessing DNA, and then we can test them by asking, are they producing the molecules which we want the new assembly lines to produce? And we can go a step further because as synthetic biologists and chemists, we're not limited to the Lego bricks that nature gave us. We can make new types of Lego bricks. We can make new enzymes. We can make new modules of enzyme activities and throw these into this design process. And so what we might get out of this is a new enzyme assembly line built from bricks which don't exist in nature, and hopefully this new assembly line produces a really powerful new antibiotic. And we call this cycle of designing things, building them, and testing them synthetic biology. This is a very powerful, emerging, highly disruptive technology that some of you may have heard of, but I can guarantee that within 10 years, everybody in this room will have heard of synthetic biology. It's going to prove a very powerful way of engineering new functions into biology. And so synthetic biology involves rewriting DNA. So for our purposes, we literally write new sequences, new DNA sequences that encode the Lego bricks that we need to assemble new enzyme assembly lines out of. And we literally just write these DNA sequences on our computers. We sometimes use software to help us design these sequences. And then we just send these sequences off to a company who synthesizes these new DNAs for us. Once we have those new DNA sequences in hand, we essentially transplant them into a microbe. And in our case, this is usually E. coli, because we're using E. coli as a chassis for doing synthetic biology in. So E. coli doesn't really make any molecules that are antibiotics or complex in nature at all. So it's like a really clean background to test our synthetic biology expertise. And so with the DNA inside the E. coli protein synthesis machinery can read that DNA sequence and make the new Lego bricks, new enzyme assembly lines that we wanted them to make. And then with these new microbes in hand, we can feed them sugars or other feedstock compounds and ask whether they're now producing the more complex products which we've designed these microbes to make. And if they do make the desired molecules, we can ferment these microbes in a very, very large scale, ultimately in industrially sized bioreactors, so that we can make really, really large amounts of these exciting new molecules. So synthetic biology is a field that's still growing and learning and developing, but there are already some really fantastic applications of synthetic biology that have permeated through various industrial markets. One is designing yeast as factories to make hydrocarbon building blocks. So this is an example of a molecule called farnesene. This is a hydrocarbon building block that's used in many, many different sectors of chemical synthesis industries. Farnesene is a building block for cosmetics, pharmaceuticals, polymers, all different kinds of things. And Amyris is a company in California that used synthetic biology to create a strain of yeast that can now produce farnesene with efficiencies that are better than other approaches to make this really key, highly important building block. And this is a very reliable supply of a key industrial building block. It can be provided with a very stable price, and it's green, it's highly sustainable. We don't have to rely on cracking petroleum or anything like that. And there are many other examples in industry where synthetic biology has been successfully used to produce building block chemicals like this one. What's emerging from synthetic biology are the efforts from my lab and others, where we're using this philosophy of engineering biology to construct molecules which are quite a bit more complicated than these chemical building blocks. So what will emerge in my lab in the next few years is the ability to diversify antibiotics like this one and to potentially discover new powerful antibiotics. Where we want to be able to go in the future is to apply synthetic biology to produce molecules that are even more complicated than some of the ones I've shown you today. This structure is called mitotoxin. As far as I know, this is the most complex molecule that's produced in nature. This contains 30-odd fused rings, dozens of stereocenters. It's an incredibly complex small molecule, which is very, very difficult to produce using traditional organic chemistry. It's the hope of synthetic biologists to engineer microbes to make molecules that are even as complicated as this. Looking perhaps even more forward into the future, what we want to use synthetic biology for is to, for example, engineer our gut microbiomes. The microbiome is something that's becoming very popular right now. The microbiome describes all of the microbial flora inside our small and large intestines and some other organs. And the way that our microbiome produces molecules and interacts with our body is really, really critical to our health and well-being. In the future, we might be able to use synthetic biology to essentially create artificially engineered microbiomes and use them as a personalized pharmacy, which essentially produces a pool of drugs inside our bodies perfectly matched to suit whatever health requirements an individual might need. In another example, we might use synthetic biology to create new microbes which act as seek and destroy elements that constantly live inside our bodies and can detect various disease states and actually move towards them, locate them, and eradicate that disease. This might sound like science fiction, but scientists are already thinking about ways of doing this, and I expect scientists to find a way of doing this in the next 10 years or so. So that's just an idea of what synthetic biology is, how we use it in my research group, and I think where this technology might go in the future. I want to finish up by thanking all of the graduate students in my research group that do this work. Some of them are actually in the room here today. I'm not able in the short time frame of this talk describe all of the specific amazing discoveries that my graduate students are making, but trust me, they're using your tax dollars to very good benefit, and they're working very, very diligently to make new antibiotic structures. I'd also like to thank the NIH and the NSF and some NC state funding to fund research in synthetic biology in my lab. And finally, I'll just include a few slide credits for those of you that might be watching online and want to see where I took some of those images from. And with that, I'll thank you for your attention. Thank you for coming out, and I'll be happy to answer any of your questions.
All right, let's give him a round of applause. Thank you, Dr. Williams. Fascinating stuff. Taking biology and then engineering it, almost hijacking it to suit what ails us, like antibiotic resistance, but even looking forward in the future and the really cool and incredible things that scientists are already pondering, ways that we can use this to make us better. So let's go out. If you've got a question, find me or find Katie in the blue shirt, and we'll bring the mic to you one at a time.
The end. Okay. No. I love all of the graphics to watch how the assembly line is working. And what always kind of fascinates me is how do you actually know what's happening? So, because you don't have a little graphic in your lab that's displaying the color-coded Lego blocks. So when you're doing this work, how are you actually tracking what's happening? You know, I know that at the end you can do a test to see did you end up with the chemical you wanted. So I guess you could assume everything went right. But how do you know it's going right?
That's a very good question. I could spend hours answering this question for you. I guess to start with, we don't always know what's happening during this enzymatic assembly line. We don't always know that it's working as we want to, but your point was correct. As long as we're producing the molecule which we want at the end, sometimes we don't care whether that assembly line is working as it should. But a number of projects in our group and in many other research groups are very interested in designing experiments to specifically ask how those molecular machines work. The video I showed you is not a real movie of how the enzyme is moving during its catalytic cycle. It's essentially an ensemble, an animation provided from years of looking at protein structures and figuring out how different states of the enzyme might be different during different portions of the catalytic cycle. So we can use X-ray crystallography to figure out some of the snapshots of those enzyme motions. We can also use a variety of chemical probes to figure out where molecules are attached and at what point of the assembly line they're involved with. So there's lots of different ways we can probe these systems and learn about them. But there's much, much more to learn. I think we've really only touched the tip of the iceberg with understanding how those enzyme assembly lines work. And some research groups are entirely devoted to learning how do those enzymes catalyze the selection and assembly of much larger molecules. It's a fascinating area.
Thanks for a very, very interesting talk, really quite provocative. One of the things that I found valuable about your presentation is your appeal to metaphors to help us understand, like the idea of an assembly line building on a chassis. If we think of the history of cars, they came to be made on assembly lines not only for technical reasons, but for other reasons that have to do with labor control, the cheapness of labor, and the like. And at one time, as I understand it, we thought of, or they thought of, making cars through something that was much more like a molding process where materials would flow into a mold rather than being assembled. So I guess my question is whether molecules are come to be in other ways than by being assembled, so that we don't get in, if they are put together in ways other than by being assembled, we shouldn't fall into the trap, which we are in now with cars, of thinking of assembly as the only way that this can happen.
It's a very insightful question. Let me start by just admitting that I don't actually drive cars. I don't have a driver's license. And so that car analogy is very challenging for me because I really don't know anything about cars. But as pictures do a good job of explaining a lot for me. Having said that, the focus in my presentation was to highlight how many small molecules in nature are constructed in this assembly line fashion. But in fact, as you've surmised, there are a large number of molecules in nature which are not assembled in an assembly line fashion, but are constructed by gathering lots of small molecule building blocks and almost folding them all up into a single active site of an enzyme where the chemistry takes place. So there's lots of other different strategies that nature has found to efficiently synthesize small molecules. What's very alluring with the assembly lines is their incredible efficiency, the speed at which they can make new molecules, and the way that we can easily envision manipulating them to make new versions of those molecules. I could give an entirely new talk on the types of enzymes and pathways that make other kinds of products in nature. I'll come again next year.
So the fine people in Hollywood have given us lots of documentaries that assure me that right now in a hollowed out volcano somewhere in the Pacific, there's an evil Dr. Williams stroking a cat, and all the graduate students are doing something, probably lasers on sharks. But what are the risks environmentally, in terms of, what happens if we go the wrong direction with something that we found, in terms of either accidentally letting this stuff into the environment, or finding out later on that these new molecules don't do exactly what we thought they did, and may be damaging. Are you building microscopic kudzu to grow across the landscape? What are the risks? What do you do to prevent the risks? And how can you feel certain that you're not the person in the hollowed out volcano creating the crazy stuff?
Another great question. Firstly, there are probably much better qualified people than I to discuss the ethics and morality of conducting synthetic biology in the future. But I think that as scientists continue to use their imagination and push the boundaries of this technology, it's really important that we continue to integrate with the public and with regulatory bodies and governments and funding agencies and everybody, basically, to assure folks that we're not all evil scientists, bent to destroy the world. At the same time, synthetic biologists are already thinking a few steps ahead, and they're figuring out how to sort of build in fail-safes and kill switches in the artificial organisms that they create, so that if they ever are accidentally released into the environment, they don't have a hope of surviving, and they'll just die. So there's lots of technology in this area that's been developed and pursued very, very rapidly. But a lot of really great thinkers in this field know that we need to convince everybody that we're doing things in a safe and careful manner. And maybe there'll be some small accidents on the way, but I don't think they're going to be anything significant. We're not going to be releasing engineered microbes that could destroy humanity. They're actually already out there, unfortunately. Yeah.
Right over here.
In looking at your slides here and thinking about the numbers, I would assume almost an unlimited number of possibilities exist in your research. You've got a oxygen here, a hydrogen over there, a weak bond here. It could go on. I assume you don't just try everything that kind of falls out of the test tube. Are there ways that you look for patterns and can say, all right, we want to go here, or we're going to try over here? Is there a way that you do that in some sort of a... You're nodding, so I think I can quit. Okay, good.
That's another good question. So, I like to describe these enzymes and assembly lines as Lego bricks that we can recombine in an unimaginable number of combinations. In reality, it doesn't work as well as that. So many combinations of those Lego bricks will never produce the molecule which we desire. And there are some other limitations regarding the exact kinds of functionality, chemical functionality that we can introduce into those antibiotic scaffolds by using the enzymes. But still, with those caveats in mind, in principle, we could make hundreds, if not thousands, of new molecules. We would love to make that number of molecules, but I think we need to be a bit smarter, as you suggested, and try to figure out what kind of new structures we should be arriving at. one way of doing this is to use computational tools to model how those antibiotics interact with the target that's responsible for the biological activity of that molecule. And by predicting how that molecule interacts with its target, which is often a protein, we can be a bit smarter and kind of home in on regions of the small molecule structure which we should focus our diversification efforts to. But largely these are molecules which are very difficult to modify by other means, and so there's little data to guide us. We're kind of stepping into new territory to some degree.
Hi, how do antibiotic-resistant strains of bacteria form in the first place? I know that's a loaded question, but...
Oh, no. Antibiotic-resistant bacteria evolve because we make antibiotics and we feed them into the environment. So it's a kind of catch-22 situation. We need to fend off infections by dangerous microbes and other bugs. And to do that, we can kill them off with antibiotics. But nature always finds a way to survive and overcome whatever we throw at it. So when we put antibiotics out into the field, eventually resistance will evolve through evolution, and that's something that we need to figure out how to overcome. So, I'm in a chemistry department. I'm really interested in learning how to make new chemical structures that are hopefully antibiotics. I think at the same time, we all need to really think about how we use antibiotics. Should doctors always prescribe antibiotics, even if it's actually a virus that's causing your flu-like symptoms, for example? There's also a big push at the moment for designing antibiotics that are highly specialized against very specific types of infections, so that we're not just killing all different kinds of microbes when we administer an antibiotic, but we're really focusing the action on one or two strains of bacteria. Is that what you were getting at?
I have a question of how do these new molecules travel from your discovery of them to our use of them?
So that is a very, very lengthy process. So we're at very early stages of learning how to use this technology to produce new antibiotics. But from the moment that we might find a molecule with a promising activity to that molecule being administered to patients might take at least 10 years. In reality, perhaps closer to 20 years. And at the moment, part of the reason why it takes so long to develop a new antibiotic is it's very difficult to construct these new antibiotic structures. And so we hope that technology like ours can be used to much more rapidly develop and optimize the structures of those molecules, so we can try to reduce the length of time it takes to produce new antibiotics that can be used in the clinic.
I was wondering how intellectual property and issues around intellectual your research, and especially the production of some of these new organisms and some of the new molecules. I think back to Dr. Salk and the issues around infectious diseases in the 1950s, and we wiped out polio, or came close to wiping out polio, because intellectual property by someone's choice didn't enter into the issue. But today we have corporations and the need for research grants and all this sort of stuff. The money's got to come from somewhere, and the way we seem to be handling that is through intellectual property rights. Is that something that's shaping synthetic biology, and will it be an issue in the future?
Yeah, so the intellectual property landscape of synthetic biology is absolutely an area which is shaping the industrial side of this technology. For me, as a professor at a university, my primary goal is to increase our knowledge and learn how to do things and make academic discoveries. But a lot of the work that we do is highly relevant to these other industries that potentially might be interested in licensing our discoveries and giving us money to do this. So, our group is actively involved in carving out intellectual property and trying to sell that to other industries. And some of these other industries, they really battle each other for rights to produce various products and to pursue different technologies. A very prominent example right now involves the use of a genome editing tool called CRISPR, which is really getting a lot of attention in the media right now. There's probably a dozen academic research groups who are all fighting for the rights to sell these genome editing tools to companies, and it's really quite a dramatic story, actually. Mm-hmm.
So I have a thought. When I was watching the visualization, that video of the prosthetic arm reaching down into different sections of everything that was going on there, and you even suggested it, it's really quite complicated. The diagrams lay it out pretty simply, and the idea of an assembly line lays it out pretty simply as well, but it looks really tough to get what you're after. How often do you come away from this process not having received your desired outcome compared to how many times you come away and you say, we got it. Good work, everybody.
I don't know that I can quantify how often this works, but...
Did everyone see the grad students in the front giggle? Okay.
I can tell you that when it does work, we're really, really happy. Yeah. But you're right. There's so many moving parts and complexities to these systems. What we need to be able to do is test many combinations of these Lego bricks. We need to test many variants of these assembly lines, which ones are doing what we want them to do. And this is actually something that we spend a lot of our time trying to figure out, is how we can identify very quickly those new assembly lines which are doing what we want. And we've actually got some intellectual property in that area, which we're hoping to license quite soon.
So knowing that right now you're essentially at benchtop scale, at NC State, are you uniquely positioned to be able to take that into, say, a pilot production or even production scale, or is that something that you envision having to collaborate with other institutes, biotech companies, pharma companies?
Another great question. This is phenomenal. So, yeah, we're doing benchtop fermentations essentially right now. We do currently collaborate with chemical engineers at NC State, where we hope to be able to produce these compounds in fermentations which are about 50 liters in volume. That's quite a high scale. That should give us a few grams of useful material. But to transition to something that's really industrially relevant, we're ultimately going to have to collaborate with industry to do that. And so that would be the next stage of evolution in our lab, is to start to think about translating what we do in an academic lab to actually working at an industrial scale. Mm-hmm.
Right here toward the front. Sort of getting back to Deb's question and the question of how often are you successful? Do you presumably do you use random mutagenesis, and might that be a means to increase the success if you can find a way to find the bug that's making the correct product, maybe by some sort of mass spec technique on a plate? Are you looking at that kind of technology?
Absolutely. So in addition to sort of reprogramming these assembly lines by introducing new components and changing the order of them, we also mimic the process of natural selection by randomly mutating the genes that encodes for those proteins. So this is a method called directed evolution, or evolution, as you might say. And it's extremely powerful, as you suggested, at improving various properties of things that are otherwise very complicated to engineer. And so by introducing many random mutations and by screening those libraries for enzymes which are now better in some way, we can completely bypass our very naive understanding of how those biological components work and basically fish them out and then figure out what they're doing. So this is something that we do every week, actually. Yeah.
All right. I think we have time for one more question right over here by Katie.
The idea of complexity has come up lots of times in your talk and in the questions. And when things are really complex, they require coordination. So I guess I have, which in coordination I understand to be primarily or essentially about sequencing and timing. I guess my question has two parts. One is, how good do you think you are getting to be at coordinating these complicated assembly processes? And then the other part is more, maybe more philosophical or even theological, and that is, how do you feel confident that the coordination process that you observe in nature is just the result of evolution through a kind of random process of mutation and selection?
Yeah, so the issue of timing and coordination is very important for understanding the molecular motions of these assembly line components and how these machines operate very, very efficiently. I think that in a very small number of very well-studied assembly lines, like the one I showed you a movie of, we have quite a good understanding of the timing and coordination during those catalytic cycles. But even if we understand how that works in that natural assembly line, if we were to start engineering it and trying to make it do something else, I don't think we know how the timing and coordination works. And so even when we do this synthetic biology, where we make lots of new Lego bricks and we evolve the system, where we pull things out that are working, we sometimes have no idea how these evolved and engineered, artificially created devices are doing their job. And can you remind me what the second part of your question was?
A random mutation and selection process that you observe in nature, evolution, can be understood to be responsible for coordinating this complexity?
To some degree, yes. So I think nature has also realized that it can reshuffle the bricks that form these assembly lines. So we can see evolutionary evidence through gene sequencing and protein structure analysis that nature has already done this. This is how nature has provided the variety of molecules that I showed you in one of those first slides. But when we do this, the general strategy might be similar to how nature arrived at these very complex solutions, but the exact things that we pull out might be completely different. Mm-hmm.
All right. Thank you very much, Dr. Williams. Give him a round of applause. Yes, absolutely. Thank you. So in 10 years, like he said, we're going to check back in, and this is the guy that we all want to know right here. Thank you so much for coming out. Thank you, everyone, for coming out this Thursday night for the Science Cafe here in Raleigh at the North Carolina Museum of Natural Sciences. Let me remind you that next Thursday night is the first Thursday of July, and that means science trivia. So get your team together, show up right here next Thursday night, and test your science smarts against everybody else. And we'll see who comes out victorious. I hope that sounds like fun. I'll be there, so I hope to see everyone there next week. Thank you, and have a great evening.